as with the autologous CAR Ts. I think we've all realized that regulatory approval is certainly a laudable milestone, but certainly not the end goal. So I'm super eager to see how those get adopted and rolled out, and the commercial sort of scale and market penetration goes. But I think lots of things. It's nice to have things to celebrate, right? Along with the downsizing and the rationalization of programs, in cell therapy, which I think has been overhyped to personally, in my own opinion. But
I do think it's interesting the way one discrete event can be pointed to as like the end or then the beginning again. I was just at
right,
BioCom and we kept hearing green shoots, right? The biotech winter is over. We have these little green shoots coming out of the earth. And then all of a sudden, boom, sunflower. Here's a $7.8 billion acquisition of a company. When like last year
right,
it was, Takeda's shutting down cell therapy. I was like,
Yeah.
what? It's like
yeah.
I'm not ready to
science
call
is
time
still
of
moving
death.
forward. Yeah.
Yeah, yeah. And I think, the other thing that we often forget is so many of these decisions, just like, any decisions in our lives are a function of a lot of factors, right? Um, you know, Companies aren't making decisions about just cell therapy in a vacuum, right? It's in the context of a lot of other things going on in the company, in the marketplace, in their indications, in their board, you know. So
Yeah.
yeah,
And
it's
I do
interesting.
think if we fix the fundamentals of the industry, as one of the things we will be extensively talking about today, that's going to reevaluate a lot of these decisions, right? Like I have a feeling they're sitting around the table going, the manufacturing's hard, it's too expensive, we have all these antiquated tools, it's just not moving forward the way some of these other fields are. And so, yeah, maybe we can
Well,
solve
and
some of those developer dilemmas.
we have so many hype cycles, right, that the industry and investors like to ride. And so everyone's looking for that next shiny thing. But to your point, you can't run to second base if you haven't tagged first and if you don't know how to run
That's right.
the baseline to get to first. But everyone's trying
Yeah.
to go for that, let me just hop forward to third base. You can't do that. If your fundamentals are broken, you're just not going to get
Yeah,
there.
absolutely. There is no doubt that the deal pipeline has slowed down, but there's really good reason for that, because we have to prove that we can surpass 20% market penetration, right? And really deliver these therapies to a much higher percentage of the patients that need them before investors are going to reward us with, further products in the pipeline, right?
Right.
I mean, God forbid we should actually have a proven effective autoimmune indication approved right now, because we're not ready.
Yep.
Right? And there's always a shiny new thing, and certainly in vivo is one of those shiny new things right now,
Hmm.
right?
Yep.
Well, but I
And
think
it,
even with in vivo, people are forgetting that, you're gonna have to solve the same problems had to solve with cell therapy. Cell therapy works great for liquid tumors and autoimmune. Solid tumors are hard. What about in vivo is gonna make that any different? We still
right.
need to fix the biology, the product
It's
design,
it's still
Hmm.
biology. Yeah.
It's still biology. Yep.
Yeah, and people tend to think binary, black and white. Well, now everybody's just going to do in vivo because it's easier.
Yeah.
Well,
Nope,
Right.
probably it's not that simple. It's not going
it's
to
not.
replace everything.
And in
Goes
fact,
back to
you're
the,
going to see a downturn because people are rushing into this with poorly designed vectors. We're going to have safety issues and you're going to see it slide, which is
yeah.
unfortunate because there is a lot of promise there. But we're in that cyclical, what's in fashion, quick to jump on and off the wagon. Yeah.
And
Hmm.
it's because people are investing in business models, not science, right? They love it because they love the business model. And there's some early indications of very promising science, but the size of investment is not correlated with the stage of the science and the data we have right now, right? So
But that's where
they're
I think
bound to be disappointed.
But that's where industry needs to leverage academics more, right? Like they're the ones that are interested in this science. So how can we support them or train them to make sure what they're doing is more readily translated to industry, right? I mean, a lot of the cell lines are vector designs or whatever you might have you started in academia, and there's issues with them, right?
Right.
And now we're trying to get around them. So I think having closer connections with academics too is going to be key for industry
Yeah.
consortiums. Those are what we really should be leveraging.
Georgie, are we live?
Yes, you are. Yeah.
All right. Well, let's all get started. Welcome everyone to this, I think, eighth session of the Advanced Therapies think tank. For those of you who are new here, this is a live session. Will be available, as all our sessions are available, online, on demand, 1 or 2 weeks after the live session. Advanced Therapies think tank is the creation of myself, Sven Keeley, and Michelle Boxall, designed to bring the fireside chat, the panel discussion that we see at conferences, and I think are really some of the most primary learning opportunities, to an online audience, to democratize these conversations and to really be provocative conversations, intended to be discussions amongst experts, who leave their corporate titles at the door and have frank and open discussions, largely, hopefully, focused around solutions, obviously to problems facing today, without really the constraints of necessarily the paradigms and precedent that sometimes we think we're forced to adopt. So with that, I'm delighted to welcome our panelists here today. We have our returning faculty member, Nina Horowitz, along with invited guests, Bashak Clements, Jessica Tate, Priya Baraniak, and Alex Sargent, who goes by Sarge. So thank you all for joining us today. I'm super excited to be here. Today's discussion, I will warn everyone, will appear perhaps somewhat scattered, but it's intentionally so. It's meant to talk about some of the primary inputs for cell and gene therapies that we're using today, and some of the primary challenges and trends in those that we can then in future sessions dive into more specifically, which we're excited to do. But before we get into the discussion with our panel, I want to take this second to thank our sponsors, without whom, of course, none of this happens. First is the BCA Advanced Therapies Network, who supported AT3, as we call it, throughout the season, and BCAR, the Blood and Cells Advocacy Register or roster, who has sponsored this particular session. And BCAR is an interesting communications and policy consortium representing stakeholders across the blood and CGT ecosystem, working to strengthen the narrative around the foundational role of blood and blood-derived cells in enabling advanced therapies. The shared experience of the members closely connects to this discussion, of course, where starting material choices shape both feasibility, scalability, and ultimately patient access. And BCAR is really focused on bringing attention to the infrastructure and collaboration required to deliver these therapies and support conversations like this. So thank you to BCAR. So with that, I'd love to get launched. I am seeing some kind of weird screen. I don't know if everybody else is seeing that. Maybe it's just me. I'll just alert you to that, Georgie. So the first question I want to start with is, what are advanced therapy starting materials, and are vectors included in it? And I think we might have a poll as well going out to the audience members on this. And I'm just going to open it up to the floor. I suspect the answer is maybe, like all complex questions, is it depends. But have at it. Who wants to go?
I'm going to claim liberty of this since I'm the vector person.
Excellent.
Yes. In the fundamental idea, they are a starting material for ex vivo, right? In vivo, completely different story. But for ex vivo, they are a starting material. However, right, because of not just the criticality, because there's a lot of critical starting materials, but the complexity of them, we can't treat them like any other starting material, which is also why then we start, deploying kind of drug substance principles to them, where in regards to the quality control strategy, things like that. So you kind of have to be able to wear both hats when you're talking about vectors, particularly those that are biologically derived, right? Because those are the most complex. and complicated to manufacture. Happy to
Just
hear what my cellular counterparts think.
I think that was a really good intro. I'll complement what you said, and I think it would be helpful to kind of level set on the regulatory descript definition of a starting material, and then kind of frame the vectors within that context. But I think a starting material is defined as a material used in the manufacture of a medicinal product that is incorporated into or are critical for the generation of a drug substance or the final drug product. And in the CGT context, this explicitly includes human cells or tissues, vectors, plasmids, and cell substrates. And then coming to the vector topic, like you articulated, it depends on the final modality, right? So if generally vectors are described as a delivery vehicle or a system that delivers the genetic material, it can be viral or non-viral, but the main premise is we transfer the genetic sequences into the cells, and the expression could be transient or permanent. And I think about the vector, like you said, based on criticality and the classification of the vector accordingly based on which modality it is being used. And the regulators, I think, also discern according to these criteria. So if the patient receives the vector directly in vivo, such as an AAV, where the vector is typically classified as a drug substance or an active substance, whereas the plasmids and the cell banks that go into making it are typically called starting materials. If the vector is used to modify the patient's cells ex vivo, then the regulators, there is slight a lack of harmonization. I think EU and EMA call it as a starting or a critical starting material. PMDA calls it an critical intermediate. FDA, Health Canada call it drug substance or drug substance intermediate. So I think there is this kind of unfortunate, terrible lack of terminology harmonization, and then the lack of associated quality and CMC expectations that come with this kind of lack of harmonization.
Alex, do you have a horse in this race?
So my horse is always, I consider vector a drug substance. And part of that is I'm based out of the US. A good deal of programs I deal with, are either trying to launch in the US market or at least access the US market, as you can imagine. So I highly advise at least thinking of it as a drug substance for now. And I can tell you from personal experience, trying to make the claim that it's just, a starting material has bitten me more than once in terms of IND submissions and the like. Now, that said, I agree there is a need for harmonization, both in the description and in the classification of the material across, all of the different regulatory agencies. But for now, I advise you take the most stringent, that you're trying to get into the market for, and you apply that when you're thinking about CMC and about the design of your product. So for me, unfortunately, I'd have to disagree. It is a drug substance.
Excellent. Off to a great start. I love this. Built for good disagreements. I think that the follow-on question is, perhaps the more important one, although standardization around this kind of nomenclature is really critical. I think it can't be underemphasized, the importance of words and classification, for standardization, harmonization, and even for people to understand what they're developing and investors to understand what they're investing in. So I don't want to underemphasize that, but we see at the bench level tremendous amount of innovation around all kinds of different car constructs and ways to get things into cells. And yet when you look at the pipeline, we're really heavily still dominated by the traditional vectors and the way we think about vectors. Is that just my perception, and is that a misperception, or are we lagging behind in our vector engineering design and selection strategies for what we're trying to accomplish clinically?
Yes and yes. So and Alex, I'm curious to hear your opinion on this, but again, coming from a viral vector specific, right? So I've worked on in vivo, ex vivo applications for vector for the last 12 years, and I can't tell you how many products now, but yes, in the sense that there's still a lot of innovation that we need, absolutely. And I think what you're seeing is as our analytics are evolving and we're starting to really understand these vectors more, and I'll use AAV as an example because I think it's kind of paving the way. When we really start to understand what is important, how to, capsid design. Right now, we're seeing all these engineered capsids to improve specificity, targeting specificity. But we're also seeing improvements in cellular uptake or endosomal escape or whatever it might be, right? And so I think we're making minor improvements here and there to try to improve this. So in some ways, we're not staying stagnant and the progression is following the progression of our analytics. But on the big picture, if we zoom out, yes, we're stagnant, right? And it goes back to that comfort where we want to stick with something that the regulators are familiar with. Maybe we could tweak it a little bit, but it's still, in principle, something that they're comfortable with versus something completely novel. And I think that's a mistake, although I understand and I fully appreciate why we do that. But in my opinion, we need to look at what the therapeutic needs and work backwards, right? And that may lead to something we already have, but that could also lead to something completely novel. And as far as the regulatory, I think we just need to have active involvement, active discussions with them, with consortium, and just get that buy-in and feedback early. So there's ways around the regulatory risks, right? Is I guess what I'm getting at. Okay, I'm done.
Alex,
No?
Alex, I think not only because Jessica's lobbed it to you, but the next question I had was for you anyway, which is, when I spoke with your colleague earlier this week, he raised something I think that might also be a contributing factor to this problem, which is it's easy enough to come up with various ways to innovate small scale around vector production and design and all kinds of cool things. But once you start translating that beyond the well and the flask into manufacturing scalability, there are some real challenges there, and that is to some extent perhaps a cause for the lag. What's your take on that?
Yes, that's a good question. So first, you referred to my colleague. I'm filling in for Chris, who does the vector production for Charles River. And I like to say that him is wiser but less attractive brother. Relationship is very good. He makes the vector and then he sends it up to my shop, usually for the ex vivo cell therapies. And the two things that I talk to him a lot about in terms of, innovation and, scalability of vector production, from the cell therapy developer's perspective, one is cost, because if you look at a typical CAR T, a big cost component and the most expensive raw material that you're going to have is your vector. So I can automate and shorten the process and do all kinds of wonderful things, but until he works on the vector side to bring down the cost and hopefully improve the scale and titer of that vector production, right, I'm at a standstill. So it takes two. It takes both of us. And the other one I tell him is that we've reached the point, especially say in immunotherapies, where if you have a good vector, you can create a CAR T cell therapy like that. But I tell him I classify in sort of two areas. There's good vector and there's bad vector, and there's no in between. There's the kind that I get and I put it on the cells and right away they light up and everything's happy. And there's the kind where you put it on the cells and the transduction is terrible and you can spend 6 months trying to optimize it, but you're playing at a losing game there. And one of the things, that we've sort of explored is what is the differentiator between good and bad? Because I don't think we really understand the biology well, both from the vector perspective and from the cell perspective, in this case, T cells. And the other one is, how do you translate that? Because the number of times you can have really, really good at small scale, and then it gets worse and worse and worse as you go to large scale, is very challenging. And then finally, I'll say one, final thing on AAV. I use it mostly, right, for ex vivo cellular therapies, say in combination with CRISPR. And I love how, I talk to clients or developers, I want to go non-viral. I say, okay, that's great. And then they're like, all right, let's use this virus to knock in a gene.
There is, and I didn't mention that, but there's definitely this drinking of their own Kool-Aid scenario, just quote Stan Keeley, right? And I think some of that is just personal feelings, right? It has nothing to do with the science. It's like, oh, I spent all my time working on this vector, so I'm going to keep pushing that vector forward. I see it on the LNPs, I see it on the AAVs. I mean, you name it. But that is a huge element, and I do think we gotta get out of that mindset of taking ourselves out of the equation, right? This isn't about us; it's about the therapy. But yeah, that is. I'm glad you brought that up, Alex.
Yeah, yeah. Bashak, I want to bring you in on this at this stage. What do you think? It's easy, as Jessica said, to sort of throw the regulators under the bus, but it's up to us to engage the regulators, right, and show them good science and show them why the risk-based approach that we're asking them to take is such a legitimate and safe one. What are the current and evolving CMC regulatory expectations and guidance for manufacturing vectors that we think are critical to this kind of conversation that we need to have?
Yeah, thanks, Lee. I mean, I think we now have enough guidance that folks have to follow a risk-based approach when it comes to CMC. I think that's iterated across the board, across regulators. I think we're limited on the how-to of that a little bit, is what I'm seeing. Both, from the overall discussion that we're going to have, I think similar thing applies to the cellular starting materials, and we can certainly discuss those in the latter part of our conversation. But even for the vectors, based on the ambiguity we just discussed, I think the amount of rigor that needs to be put on the vector manufacturing, like Sarge said, I mean, it's typically very high. It's drug substance like. I mean, whether you call it a drug substance or a drug substance intermediate or something else altogether, the functionality remains similar, and therefore there's a high degree of scrutiny on the manufacture and control of these materials. But when it comes to then, how to exert that oversight into whether your own manufacturing site or your CDMO that manufactures this, what is the rigor of testing controls? There is still some ambiguity around these topics that I think could benefit from a more evolved and more granular guidance. I sit in the USB Expert Committee for Cell and Gene Therapy in the 25-20-30 cycle, and we're looking at some of the USB guidances from these perspectives as well. There's the USB guidance on gene-based therapies that is now being revised and being solely dedicated to gene therapy. And then we're going to separate a cell therapy guidance separately from this to try to provide similar guidance on cellular therapies. In the absence of much more granular guidance coming from the regional authorities. But that might be a couple of ways we can help the field by making it more explicit what some of those expectations are.
Right. And the takeaway there is we can all play a role in setting these guidances, right? It's not just up to regulators to figure out. This is not a top-down exercise. So here's the segue question, right? Because as you alluded, we want to get into the cell side of this topic as well. And Guillermo raised it in the chat, a nice way of putting this question. We see so often people have their preferred cell of choice or their preferred cell, source of choice, their preferred vector that they did their postdoc in, or they're just precedent of, and they use that to sort of search for an indication. Is that really the best way to be approaching this? Or, is it possible to think about what's the biology of my indication in my patient population and then go and figure out the cell and vector and other, sort of input materials? Is that it seems like intuitively that's the better way to do it. And, to Sarge's point, what's a good and bad vector may or may not be a good and bad vector for everything, right? Maybe, really a fit for purpose strategy. So I'll use that segue question to engage the cell people first, and then, and get the vector people's thoughts on that as well.
Yeah,
Well,
absolutely.
Nina, why don't we start with you?
The cell people are also limited by vectors, right? And I think this is the interesting segue is in theory, I mean, if I had wings, I could fly, right? And if I could, based on my indication, design the perfect cell that was engineered in the perfect way with 17 different things, I would do it. But I'm also limited by the fact that these vectors can only deliver a payload of a certain size. And I can't do 5 different rounds of transduction for a single cell therapy product because the COGS will be absurd, right? So the current vector tools that are available, I mean, like I'm using the same lentivirus that I was using 10 years ago in my PhD. And it's not because I love that lentivirus, it's because it's the bread and butter thing that works. And we don't want to take regulatory risk on our IND filing by using some novel vector that we have to have some brand new CDMO make. No, we want to copy and paste from every other cell therapy if possible to make sure that we get to the clinic in a reasonable amount of time. So even the cell people are a little hamstrung by the vectors, but also hamstrung by the cells and our understanding of fundamental biology. I think a lot of cell therapy development has happened in these little or big silos, whether that's big pharma or the small biotechs, and there isn't really a like Wikipedia of cells that I can look at and be like, what would the best type of cell be for this type of cancer and where can I get that at scale, right? Like we're kind of working our way forwards by trying to pick the best donors and make those at scale and then use those in the clinic, but I don't think that it's actually possible to go the other way right now. I will say that one thing that often does get ignored is that donor variability and the phenotypic variability question. And I do think for indication-specific therapeutic design, you need to be rigorously examining both the function of the cells and the phenotype of the cells. Is this a cell that's going to traffic to the right place? Is it going to make the right amount of cytokine? Is it going to kill or is it not going to kill? And so, yes, I do think that's what we would all like to do, but I don't think that anybody really has the ability to do that right now.
How different is that for iPSCs, Priya, if at all?
No, there are a lot of the same considerations for iPSCs, right? And I think that there is a tendency to think that because these cells are reprogrammed to this embryonic-like state and that we started with the Yamanaka factors, there are now multiple other factors that can be incorporated in reprogramming the cells to make different cell lines, polyclonal and not just clonal, which has been the historical way of doing things with just picking a clone and cloning that clone over and over and over and hoping that that horse takes you to the golden land and the promised land. So yeah, even with iPSCs, donor differences are important. Reprogramming methodologies, how you're culturing them, the substrates you use, every unit operation and every variable has a distinct impact on the biology of the cell, the functionality of the cell, and its progeny, and what you are going to be able to do with it. There's no one size fits all, obviously, across this industry, but I agree with you. I think we are an industry full of people who have square pegs and we're running around looking for those round holes we can jam them into or those square holes we can jam them into rather than doing it the other way. Jessica brought this up, starting with the end in mind is really important and then working backwards to engineer the starting material and everything that you need to accomplish that end and not finding out when you get to phase 3 that something that works small scale in phase 1 and that horse you bet on just isn't going to get you the full way there. To Nina's point, I think some of it, yes, is worrying about the regulators and the path that we have to get the approvals and being afraid of being on that cutting edge or that bleeding edge of doing something new. But I also think it's sometimes we tend to be a little bit intellectually lazy, right? And it's because there are so many different stakeholders. Yeah, I threw that grenade because there are so many different stakeholders as well in this industry. So it's not just as a scientist what I want to do, then it's, well, how is my quality team going to feel about having to qualify this new methodology, this new material, and my analytical development team and my assay development team, and then my procurement team? Now I'm asking them to go out and qualify new vendors, and they don't want to have to do that. I mean, it's a snowball effect, right? And so I think we need a lot more courage to be able to say just because we've done things a certain way and it's worked doesn't mean that it's the best way to do it. And if we're really going to succeed as an industry, democratize access to these therapies and deliver them, we have to start with just stopping to drink our own Kool-Aid, admit where things are broken, and then say, all right, well, let's roll up our sleeves, do the hard work, and figure out how to do it better.
I think you summarized it beautifully, Nina, Priya, and I have a few things to add, but I want to turn it around and then maybe ask a question as to how we do it better. But around the aspects that we discovered, I mean, I fully agree. We are really limited by the manufacturing ecosystem and what is easy at fingertips, and in terms of who can manufacture, who can assay, and who can analyze these things. Regulatory precedent, I think, is huge, and I think there's a shift from the regulators to encourage the tried and true rather than going through these novel pathways, just so that it is reproducible and well or readily accepted. The IP and the freedom to operate remains a big boundary to some of the entry of some of these newer technologies, perhaps, and who has it and who uses them. And then the intensive time to clinic. I mean, I see that time and time again too, as a huge barrier to innovate. We're just trying to get there quickly, and there's no time to explore. But when it comes to the biology, and the question I wanted to maybe discuss further with the panel is, how do we do this better aspect? I mean, in terms of the selection of the ideal gene editing or the vector platform and the biology that we're trying to solve, I mean, I typically kind of think about, when you choose something, you think about, the integration behavior of your vector system. Do you want it to be permanent, transient, and the persistence of that system, the payload capacity of the gene that you're trying to deliver, and then the compatible cell types. And are we saying that maybe folks aren't necessarily kind of going through this logically enough to make the right decisions, or is this not enough tools in our toolbox to kind of enable a better alternative to what we're currently choosing?
I was saying the latter for sure. I think that maybe one of my biggest disappointments over the last few years is I feel like now that we have AI-enabled protein design, we should be able to make vectors that are bigger and that target different cells better for ex vivo manufacturing as well as for in vivo manufacturing. And I haven't yet seen some new splashy, huge payload capacity optimized vector for HSC or NK transduction that like people are using and showing great transduction efficiency. Like I want to put it on my cells and also see them light up, right? Like that's my goal as well. And I would have thought that that was like all of a sudden, every couple weeks, it's like, hey, we made a new vector, because I've seen it in the antibody space and the mini binder space and all of that stuff. And vectors are just also proteins stuck together, right?
Sarge, there are bigger payload mechanisms being developed now. Do you have a line of sight into those?
Yeah, I think, to that question, sometimes the industry is a bit like a snake eating its own tail. It goes back on itself. And so if you come to me and you have T cells, you're like, I want to do some genetic modification, I got options. And boy, do I got options from nucleases, transposon systems, good old-fashioned vectors, up to lipid nanoparticles, which I'm having tons of fun with. But yeah, if you come to me and you're like, I have an NK cell therapy, you're going to be like, retroviruses, spinoculation, feeder cells, what are you talking about here, Sarge? And I think that's the problem is that we tend to innovate in the areas that show the most, profitability, where we need to also think about innovating in other areas which, may not have proof of principle yet in the clinic, but do have tremendous potential. And I speak that not only as a biologist and a researcher, but as somebody who hopes to get treated by these therapies one day, that we just can't be short-sighted on that.
Right.
Careful what you say. I think NK cells have great proof of principle. They just don't have big venture money. And that's an important
I've
difference. But
seen
I
them
agree with you.
die
It's,
and
it's
come
all
back
hype. Yeah. Yeah.
time and time again.
Yeah.
Let me use that to launch into something that we decided was important to talk about, and Priya touched on it a little bit, but and that is one thing to think about the different functions of the cell and what we understand that they typically perform, and the different functions of the vectors and what we want them to accomplish. But the one thing I think, particularly for allogeneic cell therapies, that has been an understudied science, certainly under discussed, is all of these things, the cells at least, now we're transitioning to the cells, come from donors. Even if you're creating a cell line, and there's a huge amount of donor variability, and there's a couple of shining examples, I think, of companies who have done a phenomenal amount of work on donor characterization and trying to ensure that they are not, subject to super donors that are impossible to find. and yet also finding enough, understanding what donors they really need to find to have an adequate program. And one of those is with us today, Nina. Is it ever acceptable to say, we just use young, healthy male donors?
I think that's intellectually lazy. Like I said, that's the best thing we had for a long time, right? Was younger is better, fresher is better. But we have to, again, really, I think, interrogate those assumptions and see if the functional outcome, the biological outcome in the patient population really correlates to those young, healthy donors. And even with young, healthy donors, there's so much biological variability, right?
Right.
If you choose a male, okay, great. And maybe one of the reasons we did it was also because our clinical trials have largely been developed, or, designed around men and treating men, and our therapies are designed for doses that are for men mostly. But we're trying to change that again by being more inclusive and being more thoughtful in all of this design. And so our starting materials have to also reflect that variability. We have to develop processes that understand that variability and can withstand that. If your process is based on, I need to apheresis someone, I like to say, who ate 11 kernels of corn last night and jumped on 1 foot for 22.2 seconds, that is not a commercially viable process that you have. So you have, heterogeneity is really important for a number of reasons, and building that into your process development and manufacturing programs, I think, is very, very important as well. Some of the best donor cells we've seen, in my experience, some of them have come from 60-year-olds, and the 20-year-old donor cells have not performed as well. And then there's something also to be said for the memory of various cell types, not just immune cells and T cells, but epigenetic memory of other cells as well, right? Originally, we thought you could take a skin cell, reprogram it to an iPSC, differentiate it, and it had endless possibilities. And now we've come to understand, well, if I want a CD34, it's perhaps better to start with reprogramming a CD34 because of that epigenetic memory, right? So similarly, is healthy always better, or could it be that a disease donor's cells, because it's already used to that that morbidity or that disease state, might respond better? We're not really delving into these biological questions.
Yeah, it's fascinating. I mean, we had a conference we were in December that the donor of preference for this company was chronic cannabis users. So
I was about to bring that up. Yeah.
There is a donor for everyone. But you would think, Nina, if just choosing a general population, was the strategy here, choosing baby, umbilical source material would be the great equalizer, right? Because there's no history there. But you found differently, right?
Well, so I think there's variation in anything, right? Even identical twins are a little bit different. The closer you look, the more variability you will see. So yes, we use babies that are 0 days old, but even there, I mean, Katie Razvani published this incredible paper, and I'm so grateful to her for doing this, where she ran that clinical trial with NK cells from different cord blood units. And she saw 2 completely different efficacy curves from umbilical cord blood NK cells, manufactured in the same way, processed in the same way, same CAR construct, everything. And one of the variables that was most important was whether the unit was frozen within 24 hours of collection.
Exactly. Yeah.
So there's like the age of the baby, then there's the age of the unit, right? You want consistent processing, but even within then, so we always request units now that have been frozen in less than 24 hours, but even still, we see massive variability. And so I think every product is different, right? There was another company at that conference that wanted people who had been recently infected with COVID because they were looking for COVID responsive T cells, right? For other people, that would be an immediate disqualification. For us, I would never touch anybody's blood that's older than 1 day, right? So everybody has a different idea there. But I think we need to be looking at the clinical data and understanding. I mean, like Poseida's doing allogeneic T, right? There's lots of people out there, Caribou. I think we're going to see data coming out more and more that supports the fact that even if you have the same selection criteria, the cells coming out at the end are going to be a little bit different. You're going to have donor-derived variability. You're going to have batch-derived variability. Even from one master cell bank, you do 2 runs, they're going to look a little bit different, right? And so I think we need to be acknowledging that and then figuring out how to set your selection criteria wide enough that any patient who needs a therapy can get it. We need commercial scale.
Right.
But stringent enough that the therapies you're producing are still high enough quality that you feel comfortable giving them to a patient. And so we need to link those clinical outcomes back to donor attributes in a way that's not just young male or processed in
Right.
24 hours. There has to
Right.
be something more than that.
And as someone who represents the industry that wants to find the right donor for you and collect them in the right way and process them in the right way, we need you to just do the science enough and to team and to partner with you to do the science on that, so that once you give us, okay, go find this donor, collect and process it in this way, we can go find that donor.
But
Right?
I'll put
Because they're
another
out there.
asterisk there. Even if you need to recall the same donor, they're going to be different, right? So the other part of this is you need to find the right donor in time and space and then make enough cells from them that one time that you can continue using that to make a consistent product. And so I think recalling a donor, what if they didn't sleep that well the night before? What if they had 17 kernels of corn for this one time that you were
They
going to
went
get
on a bender, they got a
right?
cold. Yeah, I mean,
Right.
you don't
And
get
so
the
then even there you
yeah.
have this like you need quantity of your high quality cells. And that's why I
But
think people turned to iPSCs.
yeah, but I think at the end of the day, right, it comes down to our lack of understanding, right? This is all the
Yeah.
guesswork, the number of corn, the number. But I think right now we're in this position where our field is lining up with another field that we really need, which is this deep technologies that are out there, right? This high performance computing, the AI. And I know some people have embraced it, but there's a lot of people in the industry who don't have the resources necessarily to embrace this. And it's gonna take all of us coming together to share these resources, share these data, to understand how all these factors are coming together, right?
I
There's
want
millions of factors, right? And
it
And
doesn't
also,
cost that much to do functional profiling in vitro, though. Like, even if you don't have the resources to screen 1,000 donors and make a perfect profile, you can take a little bit of your material and you can really
you
rigorously
could
screen
still
it
gather
before you
data.
bring it to the
Yes,
clinic. Yeah.
yes. And
and
then
I
that's
and
where I
I
think like the consortium come together, right? Because if this person has this data and this person, yes, there will be some data formatting issues or data engineering issues we're gonna have to get around, but it's still a better place than where we were. And hopefully we can get to this predictive mechanism where we're looking for certain markers, like, oh yeah, he had too much corn the night before, right? Or
but
you need old lady, middle age, cat lady blood. And I would say, here, I'm right here. Take my, but I mean, but
but
we're at this convergence, and I think we've got to figure out how to take advantage of it better.
But the only way to make those models work, and it's just an outstanding point, is you need the data points. And to get the data points, you need to obviously to invest in the science and this point, but you need the analytics, right? And
Yes,
this is the
yes.
other under
But we can,
invested area, I think, in the sector. We need, like, we're working on a project right now with Lumicite on trying to correlate, one of the challenges that Nina's working on, which is, why do some chords flatline in expansion and some chords grow like mothers? Everybody needs, but we need better analytics to be able to apply to those kinds of correlations so that we can then have enough data to put them through those models.
absolutely, but that's model refinement though too, right? So I
Right.
don't think we should wait for perfection. Let's
No.
let's do what we can. And because I mean, I told you about this, right? Like I'm big into analytics as well. Within CCRM, we have both an analytics kind of initiative going on as well as, this like an AI kind of high performance computing initiative going on. So we're trying to tackle both at once, but we're not waiting for either to get where it needs to get.
Right.
I'm
Right.
worried
Right.
about AI, that we view it too much as a savior. I was just a part of a project where we fed, right, an AI platform. It wasn't 1,000, but it was hundreds of donors. It was like, all right, predict for me a good donor in this process. And it said, no problem. You want a young, healthy male source. I spent how much
That's
money
because
and how much time to arrive at this?
But I think it's because people don't, and there's another translational issue or translation issue, right? Because I think these are two different fields, and we're trying to each dabble into each other's field without knowing the language, right? And I think without me as a biological scientist understanding how AI works, I'm going to run into these pitfalls where it's just pulling from my own files and feeding my answers right back to me, right?
Right.
And
You have
so, yeah,
bias in your training set. Yeah.
yeah,
Exactly.
Always
Yeah.
tell
but it's just
people
like
you
our
have to be
field,
careful.
right?
I could feed that AI model, 1,000 data points of what I had for breakfast that morning, and it would correlate that somehow to the functionality of my cell therapy.
But it's
And
like saying
so,
advanced therapies aren't successful because we've had some failures, right? I think we're going to have failures
I
with
want to
in
flip
advanced.
this around though. There's a difference between generative AI and really good machine learning models, right? And so what
that's
we
very
did is
true.
we fed it
Yeah.
thousands of data points. We've screened, dozens of different donors. Every time we do it, they kill 10 different tumor cell lines, solid tumor, 4 different liquid tumor. We run all these functional assays. And then we got it so that it could tell us based on the attributes of the HSCs, the immune stem cells, how well the natural killer cells would function within our system. And so now we can run twice as many donors in half the time because we expand their HSCs and then we stop and we feed all those into the model and it says, okay, this 50% is worth advancing to NK differentiation. Do I let it make the choice of what I'm putting in the clinic? Absolutely not. But is it going to get me there faster because it can help me screen out duds earlier because it can do pattern recognition in a way that I can't, like just squinting at my spreadsheet, being like, oh, the CD34 expression, blah, blah, blah. Yes. And so I think machine learning models and rigorous statistics are incredibly useful when used properly. But yes, AI is not going to save any of us.
But again, we need these large data sets and we need to share as an industry, right? Whether it's consortia and pre-competitive models that we're using. To your point, Alex, if we're just feeding it our own data, it's going to tell us exactly what we want to hear and not necessarily what we need to know. And so we have to, these IP issues, Bashak, you brought that up, right? How do we circumvent some of these IP issues, some of this, well, this is mine. I've put so much of my time and energy into developing this that this is now a proprietary asset that I don't want to share. And what we do again and again is we allow others to make the mistakes we've already made and we've learned from rather than contributing that data and those learnings in a way that help, a rising tide lifts all boats, right? We've got to take more of
So,
that approach.
I'm going off script here, but I think this is a very good point, because there's been a lot of people talking about V2V consortium and data loops and ecosystems, and they're all well and good, but practically speaking, how can we make it work? That's the common question I get every day because IPs and lawyers get in the way and blah blah blah. You've recently built a consortium like this, Priya, and admittedly, it's relative to your work that you're doing right now at Pluristics. But can you just give us a high level of what the objectives of the consortium are and how you think it can work, and accomplish what?
Yeah, and so we have 8 founding industry partners that have come together with complementary tools and technologies. It's called the Organoid Commons. It's a consortium for the optimization and measurement, manufacturing, and optimization of a network of standards for organoids. And so to really move NAMs forward, right, especially with FDA 2.0 and other pushes we're seeing globally to move away from animal testing. And what this has the opportunity to become is another wild, wild west, as we've done with so many other things, where everyone has their home brew, everyone does it their own way. It's a bespoke art rather than true engineering, science, and industrialized principles. And we can't compare apples to apples and move rapidly forward as an industry. So we're looking to circumvent a lot of those pitfalls that we've already done time and time again. And so the way we're operating is through project calls, where different partners will come together based on what technologies, platforms, tools, expertise are needed to contribute their pieces to the puzzle and work together to either put out products that can be readily available to the industry, to streamline workflows, standardize workflows, make reference standards and materials, or just put out the workflows with bill of materials, right? That says you go here, 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 use these, do this, you get this outcome every time. And so we've all come together. There are some competing interests on this consortium with the founding members. We have an open call that's going out soon for new members, and we've had a lot of interest coming in, and there are competing interests, but we're putting these competing interests aside to move the field forward. And so, again, it comes down to the people. The organizations matter, the lawyers, of course, get involved, but if the right people come to the table with the right mindset and in the right spirit, we can do anything we put our minds to.
Yeah, I think it's a wonderful precedent, and Priya's been kind enough to share the sort of the founding documents with me, and I'm really keen to put something together, something similar together in analytics. So if anybody's interested in joining that, sort of let me know, and I think that could be quite exciting. We have about 10 minutes left. I'm going to jump to one of the bonus questions we'd identified because I think it was a team favorite. Has the industry over-indexed on cell quality at the expense of defining functional quality metrics that can actually correlate with clinical efficacy? Are we moving in that direction? Are we trending that way? Or are we still stuck in we need this many cells or this much liquid in a bag?
Priya and I
We
both
do.
unmuted at the exact same second. We like unholstered and ready to go. Go ahead.
Nina, I'll give you first shot.
Okay, thank you. Yes, I think so. And this is my personal vendetta against the use of iPSCs to generate certain kinds of immune cells. And I will bound it to that because I do think iPSC technology is great. And for some things, it's incredible. And we do need quantity. And some cells are just impossible to get from a donor. You can't pull neurons out of someone's brain to give them to someone else. You need iPSCs for that, most likely. But for certain immune cells, the quality isn't there. But people tried to make them with, feeder cells pulling from variable donors. That didn't work. And they got, I'll say, intellectually lazy and said, well, we can just make them from iPSCs and we can make tons of them and they'll be so cheap and consistent. And the quality wasn't there, but they were more focused on getting, 1 billion cells per donor and being able to make this for a certain COGS target. And so they advanced those programs forward. And I think, unfortunately, the clinical data showed that iPSC-derived NK cells in particular were not as efficacious as we would have hoped they were. And then the field got a bad reputation because of it. And that was just what happened. And I think really what should have happened is that when the quantity became an issue, we focused more on how to solve the quantity using real hematopoietic stem cells or real natural killer cells, rather than just abandoning that and saying, well, iPSCs grow infinitely, so let's just do that and throw some differentiation approach on the back end. And so what we at ImmuneBridge do is we take this orthogonal approach, which is where we take hematopoietic stem cells and then we expand them to scales comparable to iPSCs and then differentiate them. And we benchmark these. And that's the important part, is you have to benchmark your product. And so we benchmark them very rigorously against peripheral blood NK cells, cord blood NK cells expanded with and without feeder cells. And it's only once we had an expansion and a differentiation protocol that we're achieving comparable cytotoxicity that we said, okay, now we're ready to move forward with a product. But I think some people thought, as long as you can make enough cells, maybe we'll just inject everybody with 2 billion NKs and then it will work. And in reality, that's not at all how it worked. And so, yes, I think for certain lineages, people just over-indexed on quantity instead of quality, but it is the quality that determines the therapeutic outcome. Like a patient doesn't want to die, but say, well, at least I only paid $3,000 for my cell therapy. No, as we've seen from the industry, they'd rather live and pay $500,000 for an autologous CAR T, right? So you really need both. You need high quality cells at commercial quantity.
I think we're seeing it in T cells as well, right? Injecting a whole bunch of exhausted T cells. Sarge.
What if I can't pay $500,000 for that life-saving therapy?
Then you better hope that someone funded Caribou or Poseida to get your allo CAR T.
Yeah, well, we have to democratize access. Yeah. And we can't sacrifice quality for quantity, but even quantity is oversimplified, right? We are again, I think we come from such a feeling of a famine in this industry, and I've got to hoard my resources and I've got to hoard my cells, and we focus on that and lose sight of, again, that end goal. But even something like cell enumeration, and we were talking about this before I think we went live, right? There's this thought that quantity is easy. And we talk about quantity being hard from a manufacturing perspective, but to go back to analytics, people will be like, oh, counting cells is easy, determining viability is easy. Go talk to some of the people at NIST or NIH or elsewhere. It's not easy. And if you've ever tried to validate these assays, no, it's not. And if I'm counting my cells on a salometer versus a hemocytometer, which yes, people still do that, versus, an NC200 or an NC3000, I'm going to get vastly different outcomes. And so again, even at the very fundamental thing of quantifying cells, the very fundamental metric of how many cells do I have here, we've got so much variability and drift across this
Again,
industry.
Let
analytics,
alone
data
the
and
flow, right?
analytics.
Have you ever seen CD34 quantification done?
Oh
Like you
yeah,
look
I've
at
done
the
it.
flow,
Yeah.
it's like a
Yeah.
smear, and you're like, this is the gate.
I'm going to gate it this way. And then,
Yeah.
you have those people, I'm just going to drag the gate like a little bit this way. Yeah.
And Leonol, I want to chime in on the analytics piece and also touch upon, what we do with the findings of our, in-depth cell characterization, and perhaps what we can also learn from the autologous cell therapies, who are now manufacturing larger populations of patient material, and they are encountering these issues and probably and hopefully gathering more and more data to try to dissect what that means. So when you get a donor material and your cells don't grow, does that mean that doesn't grow under any circumstances, or does it not grow under your defined circumstances, but may grow if stimulated differently, for instance? So I think those are the kinds of in-depth investigations that we as a sector need to get into to be able to really utilize and maximize the choice of therapy that we start a patient journey. And I think, we're currently not doing that, unfortunately, and our manufacturing platforms aren't probably conducive to making that adaptable manufacturing decisions based on a patient cell that doesn't grow at initial try because you're stimulating it with something, but didn't try something else. It would be amazing to kind of move in that direction.
I want to take one audience question. There's so many to pick from. So such a good chat going on the side, and I look forward to reading it when I have a moment. But I'm going to pick Therese's comment and get the panel to opine. So she said, "I don't understand why we expect to have attributes of the cell product's predictive efficacy. We need to remember that it's not only the functions of the product cells that make outcome in patient; it's the in vivo environment within the patient that makes a big impact too, since the product cells interact with the patient cells in the environment." Thoughts?
I mean, that's true, very true. But I mean, there's so much population heterogeneity in most places, right? And so if we do want to have products that we can truly democratize, we do have to try to find some way of correlating the starting material to the outcome. And I do think that there are correlations, as Nina mentioned, it's been demonstrated in certain cases. But this is where I think, again, my take on it is fail fast, fail hard. Find out what breaks your system, what breaks your process today when you're in early process development before you get to clinics, so you understand how that affects those outcomes with patient
But
variability.
isn't that also just to making the case for post-infusion diagnostics and follow-up and adapting your treatment
Yes, the
regimen
analytics, of
accordingly?
course.
Yes,
But I
yes.
also think it's making the case for taking more shots on goal, maybe bringing multiple donors into a clinical trial or multiple different iPSC lines as your sources, because there are undeniably interactions between your donor and your patient that will be different. Like it might not be
Yep.
that the best donor is the best donor for everybody. It may be that you want a little bit longer
Absolutely.
persistence of your cells. So you want a donor that fits a certain cytotoxicity range, but also has a certain HLA match, right? And so I do think we can't ignore the patient side. And by bringing a more diverse cell product forward, you can really learn what are the attributes of both that are going to combine to give you a good outcome.
And on the flip side, patient stratification, right?
Yeah, but I don't want to just exclude someone because they're not HLA-O2. Like those people have enough therapies, right? Like I don't want to do that part.
Yeah, and I think Therese keeps bringing up precision medicine. That's really important, and I mean that's a whole separate conversation we could have for hours. But I think this is also where, from autologous therapies, there's a lot we can learn for allo, right? And how patients are reacting to their own cells, the modifications we're making ex vivo to those cells, and transplanting back those data sets, much like we've learned as a cell therapy and gene therapy field from what happened in monoclonal antibodies, from an allo perspective, we can learn from what's happening in auto. And we often, I think, Lee, you brought this up earlier, we tend to be so binary, but all of these things are really interwoven.
I really want to thank all of you. I think on the eve of International Women's Day, which I think is Sunday, it's nice to have a heavily dominated female panel, chosen simply because they were experts, outstanding experts in the field. This is one thing I promise, is that we will have future think tanks focused more deeply on each one of these subjects that we talked about today. I think it's once in a while, it's great to have a high level discussion teasing up a whole bunch of things, because as was pointed out several times in today's session, this is biology, and it's not as singular mechanisms of action as we like to think of, and as Western science forces us to be disciplined around, I think. So as we close, again, I'd like to thank our sponsors, BCA and BCAR, who make this possible. If you are interested in sponsoring, certainly let us know. And if you've enjoyed this session, please pre-register for future events. It's on our website, advancedtherapiesthinktank.com. Subscribe to our mailing list. Join us on LinkedIn. Be part of the community. We are really enjoying these conversations. I think we learn more than anyone. And so really love the participation of our faculty, our invited guests, and certainly if you have ideas and suggestions on future topics, we're sort of all ears and open to those as well. So thank you to the audience for chiming in on the chat. And we look forward to future discussions. Thank you. Thank you all.